How about the proteins? The proteins play many roles in our system. Now, there is one role I would like to mention that I want you to remember going forward, even though we are also going to talk about other roles. That role is the role of enzymes. Enzymes, the very essential components of the metabolism, whether you talk about dehydration synthesis reactions or hydrolysis reactions, enzymes are the one that are catalyzing these reactions. So you cannot live without enzymes. Okay? That tells you how important proteins are. Okay? So proteins play the role of enzymes in your system, and these enzymes are essential for metabolic processes that include reactions of synthesis and reactions of breaking down. Beyond that though, you have that proteins are going to play the role of support. Think for instance of your hair, right? Your hair actually plays a very important structural role in your body. Some proteins play the role of transport. Think of hemoglobin that catches oxygen and transport oxygen from one place to another place in your body so that that oxygen can be used to harvest energy from the food that we eat. Some proteins plays the role of defense. In defense, think for instance of antibodies. Antibodies are produced to provide you a protection against unwanted particles that come in our system. Okay, some proteins are going to play the role of regulation. Now think a minute. A little boy who is 5 years old doesn't have a beard or a mustache, right? But a male who is in his 20s, 30s is going to typically have a beard or a mustache or both, right? But how is that possible? This is possible because when you are a child, some proteins that play the role of regulators are going to shut down the expression of some genes that are going to be responsible for the production of beard and mustache, right? So when you grow and you become adult, or you become full-grown adult, right? At that point, these genes that were at one time turned off because of the regulatory proteins that prevented them to express, now these genes are on, right? You have the proteins played a very important role there so that you don't mature before time, right? And then motion. You have someone, running here. Without muscles, we wouldn't be able to run. Without muscles, I wouldn't be able to do all the motions and talking I'm doing here right now, right? And the proteins are the one that fuel these motions in the muscles. When we consider, for instance, the net of the web, I should say, here a spider, that web is very much made up of proteins. Okay. All right, where do we find protein? We're going to find protein in milk, in eggs, beans, nuts, tofu, in the meat. Okay, this is our major source of proteins. Now you have many other sources of proteins. These proteins now though, they are made up of building blocks that are called amino acids. Just like we saw that monosaccharides are the building blocks of polysaccharides, here the amino acids are the building blocks of the poly something that will make proteins. I'm saying poly something because for amino acids, the story is going to be a little bit interesting. Now, let's go back to amino acids. The amino acids naturally occurring, we have officially 20, but research shows that we have 21. But, you know, because this is introductory biology, I'm not going to bug you with the 21. I want you to remember the 20 amino acids. These amino acids are similar to with the fact that they all have systematically an amino group and a carboxyl group. They all have these functional groups. Then they are going to be different based on the side chain that you see here. So that side chain here is that of valine. That side chain here is that of tryptophan. That side chain here is that of glutamic acid. Okay, and that side chain here, it's that of cysteine. All right, so an amino acid, it's basically a side chain with an amino group and a carboxyl group. Now, these amino acids are going to come together through dehydration reactions. When they come together through dehydration reactions, they end up forming what we call a peptide bond. Peptide bond. That peptide bond causes us to say that we have from one amino acid, two amino acids here that are not connected together through covalent bond, to two amino acids that are connected through covalent bonds. We call this dipeptide. Why dipeptide? Because we have a peptide bond here. Okay? Dipeptide. All right? And you have the release of a water molecule, so which means that this is a dehydration synthesis reaction. Right? Now, this peptide bond can develop between multiple amino acids, so that we are going to go from dipeptide to tripeptide to tetrapeptide to pentapeptide to polypeptides. Polypeptides. All right, polypeptides. Now, polypeptides can also be called proteins, right? Everything depends actually on the size of the polypeptide, but for the sake of this course, I will not bug you with that. Now, the polypeptide, when they are newly synthesized, it's going to be chains of amino acids, right? So we have an amino group here and a carboxyl group somewhere here. So we have one amino acid, two amino acid, three amino acid, four amino acid, and so on and so forth. And they are all connected by peptide bonds here. Okay? So that necklace kind of structure that you see here constitute the primary structure of a protein. So when a protein is newly synthesized, the sequence of amino acids describes a primary structure. But then, because some amino acids are polar, some are non-polar, some are charged, some positively charged, some negatively charged, you understand that that molecule that is in its primary structure can actually develop some interactions between amino acids that make up this chain, right? This interactions end up creating foldings in the protein so that there is a secondary structure. That secondary structure fall into two categories. We have a secondary structure here that is alpha helix. That alpha helix, it's almost like a spring, right? It's almost like a spring like this, okay? Stabilized by hydrogen bonds. So the hydrogen bonds are the one that are going to stabilize that alpha helix. The second type of structure that we're going to notice, it's that of a beta pleated sheet. In a beta pleated sheet, it's kind of like an accordion, like this, stabilized by hydrogen bonds. So the secondary structure of protein consists of a spring-like structure called alpha helix, stabilized by hydrogen bond, and the second one, it's an accordion-like structure, right, stabilized by hydrogen bonds. And furthermore, more interactions and more complex folding can develop out of the secondary structure, so that the protein will end up adopting a three-dimensional shape. That three-dimensional shape, it's a mingling of the structures that make up the secondary structure, so that we have a tertiary structure. That tertiary structure typically is globular. That globular determines the shape of the protein. That shape of the protein does everything, means everything in terms of the protein function. If that protein is properly shaped in 3D, that protein is going to do the job it was designed to do in the cell. If that protein is not properly folded in 3D, that protein will not do the job it was designed to do, right? I want you to remember that. So the shape of a protein, the three-dimensional shape of a protein, is essential for its structure and function, which means that if for some reason something happens so that you have the loss of a shape, this may imply also loss of function. Whenever you have loss of shape and loss of function, this leads to a condition that we call denaturation. What are some environmental conditions that can cause denaturation of proteins? A drastic change in the pH. A drastic change in the pH can cause denaturation. A drastic change in the temperature, especially in going high, right? Going, for instance, from room temperature to 37 degrees Celsius. Okay? This temperature change, it's enough to cause what we call denaturation. Another condition that can cause denaturation is radiation, right? When we have radiations, that can cause proteins to lose their shape and function. Now, let me tell you this, and please write this down, that the tertiary structure of a protein is the minimal structure that is needed for a protein to be functional. Let me repeat that. The tertiary structure of a protein is the minimal structural level for that protein to be functional. In other words, you can have a perfect protein that is in its primary structure. That protein will never be functional. That protein can be perfect, but If it stays in secondary structure, that protein will not be functional. It will only be functional if it adopts its tertiary structure. Now, with that being said, some proteins actually, in spite of being in 3D and having all the sequence correct, will not do their work. Why? Because some proteins actually will only do their work when they complex with other proteins. That's the case of hemoglobin. One single molecule of hemoglobin is not enough to carry oxygen in our system. Two molecules of hemoglobins are not enough to carry oxygen. Three, they are not enough. You need actually 4 molecules of hemoglobin in order to produce a functional unit. Okay? In that case, we say that the protein is in its quaternary structure. A lot of the regulators that control gene expression function that way. So the body is going to control at which point this regulator is going to be active or not by just controlling how much the regulator is expressed, right? So the cell on purpose is going to keep the regulator in low amount so that they are never in the optimum ratio to do their job. Whether that job is in terms of shutting down the expression of a set of genes or stimulating the expression of a set of genes, right? Both work, okay? Finally, the nucleic acids. The nucleic acids are the core that holds the genetic information in a living system, right? So we have DNA, deoxyribonucleic acid, deoxyribonucleic acid, DNA. That DNA is the molecule that carries the information, the genetic information that explains why you look the way you look. And then the second molecule that is part of the nucleic acids is the ribonucleic acid. The ribonucleic acid, RNA, it's a molecule that actually is a derivative of DNA. That derivative of DNA allows proteins to exist. In other words, we go from DNA to RNA to proteins. All right? And unless you have that sequence, you won't have productions of proteins. Now, with that being said, the building blocks of nucleic acids are called nucleotides. These nucleotides are made up of 3 parts. A sugar, that is a 5-carbon sugar, 5-carbon sugar, a nitrogenous base, okay, and a phosphate group. Now, the nucleotides occur in 5 kinds. You have adenine, guanine, cytosine, and thymine. But there is a 5th kind. That one is uracil. Now, while guanine, cytosine, adenine, and thymine are found in DNA, in RNA though, you're going to have guanine, cytosine, adenine, and uracil. Okay? So whether you are in DNA or in RNA, you're going to have 4 nitrogenous bases, right? Now, in DNA, we're going to have that DNA, it's a double-stranded molecule. A double-stranded molecule in which the strands are wrapped around each other. So this is one strand of DNA, this is another strand of DNA. And we see that the two strands of DNA here are stabilized by hydrogen bonds. Okay, so DNA, it's a double-stranded molecule stabilized by hydrogen bonds, right? And this hydrogen bonds develop in a way that guanine always pairs with cytosine, while adenine always pairs with thymine. All right, so these are some of the things that I want you to remember about DNA. Now remember that DNA contains thymine, RNA contains uracil, right? Now, how about RNA? RNA, in addition to, adenine, cytosine, guanine, you're going to have uracil, right? That uracil, it's what causes RNA to stand out compared to DNA. But now let me modify. It's not only that. The sugar in RNA, it's ribose. The sugar in DNA, it's deoxyribose. Deoxyribose. That's a 5-carbon sugar. All right. So you understand that between ribose and deoxyribose, we are talking about the same thing, except that one has an oxygen, an extra oxygen, while the other one is missing oxygen. The sugar in DNA is missing oxygen. That's why it says deoxy, right? While in ribose, it has the sugar, the oxygen, all right? RNA, it's a single-stranded molecule. RNA, it's always a single-stranded molecule, normally, right? Okay? And again, RNA, it's a byproduct of DNA in the sequence of gene expression. All right, so DNA is converted into RNA to produce proteins. Now, when DNA changes because of a mutation, that can have a tremendous impact on the protein, not only shape but also function, right? Here we have the example of the normal hemoglobin gene, where we have valine, histidine, leucine, threonine, proline, glycine, and glycine. Now, here in a modified hemoglobin molecule, because of a single modification in nucleotide in DNA, we have that glycine is replaced by valine. That single change that result in the change of one amino acid with another amino acid has a tremendous impact on the red blood cells. Now, here we have a biconcave red blood cell, a normal red blood cell, right? Here we have a sickle cell that is nowhere biconcave, right? This sickle cell, as you can see, the shape is the shape of a sickle, right? So this sickle cell have a hard time navigating small blood capillaries. As consequence of that, you have multiple complications in individuals with sickle cell disease, okay? All right, so the nucleotides which make up the nucleic acids are also used as source of energy. ATP is such source of energy. ATP stands for adenosine triphosphate. So adenosine triphosphate. So this right here, it's adenosine, and this is the triphosphate. So we have 1, 2, 3. This molecule is the currency of energy in our body. Why? The reason, it's because this is the simplest molecule that gives off energy easily in our system. Why is this possible? Because this links here between phosphate are filled with energy. Now, let me attract your attention first of all upon the fact that you have OH, 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 OH. So remember also that oxygen is highly electronegative, right? Oxygen is highly electronegative. So this area, this tail, right, is going to be relatively unstable. It's going to move extremely. And because we have a bunch of negative charges here, they're going to repel each other. They're going to try to get away as soon as possible, right? So that readily the outermost phosphate is going to leave like this, okay? So that we go from ATP to ADP plus a phosphate that is going to go somewhere attached to another molecule, for instance. Now we end up with ADP. So that right here is ADP. But notice that even as ADP, the tail of phosphate is still unstable because we still have some energy here, which means that that one can also split. And when that splits, now we go from ADP to AMP and a phosphate that get attached to another molecule. Now, what does ADP stand for? Adenosine diphosphate and adenosine monophosphate, AMP. Okay? So, that's what you want to remember. ATP is the currency of energy in our system. All the food that we ingest and the energy that we obtain from that food, unless it is converted to ATP, that energy is good for nothing. Okay? Let me repeat that. All the food that we ingest that represent energy, unless that energy is converted into ATP, our cell will not know how to use that energy, right? It's like having a bunch of dollars in your pocket. You travel to Europe, you go to a restaurant, say in Italy, and you ask for a sandwich, and they come to your table and they ask you to pay, and you take a $100 bill out of your pocket and say you're going to pay with that. Well, at the restaurant, they're going to tell you that, well, sorry, sir, we are not accepting this money here. Well, does that make the dollar less of a money? No, but it's not the proper currency in Italy. Vice versa, right? If an Italian was to come in the United States and bring euros in his pockets, these euros do not mean anything at McDonald's or at a coffee shop, right? Such as Dunkin' Donut. That euro do not mean anything. They have to first of all convert that euro into dollar so that they can get their coffee and they can be happy. All right. So, as a summary to this chapter, we talked about sugars, not to this chapter, but to this segment of the chapter. We talked about sugar. Let me repeat this. As a summary here, the DNA structure is made up of a 5-carbon sugar that we call a deoxyribose, while as a summary, DNA is made up of a 5-carbon sugar, 5-carbon that is called the deoxyribose, while RNA is made up of ribose, right? DNA contains nitrogenous bases. These nitrogenous bases specifically are adenine, guanine, thymine, and cytosine, while in RNA, we have adenine, guanine, uracil, and cytosine. In terms of strands, the structural organization, DNA, it's a double-stranded molecule, right? With adenine pairing with thymine and guanine pairing with cytosine, while RNA, it's a single-stranded molecule. DNA, it's a helix, right? While RNA is not. Okay? Now, let's take a look at the molecules of life. The molecules of life fall into 4 categories. The carbohydrates, in which we are going to have the monosaccharides, disaccharides, and polysaccharides. The function of the carbohydrate is going to produce short-term energy. Some of them are going to play a structural role. Okay? Then the next category of molecule, it's the lipids. Among the lipids, we're going to find the fats, oils, phospholipids, and steroids. They are all characterized by the fact that they are water-fearing, right? Okay? We're going to have the fatty acids, we're going to have glycerol, we're going to have cholesterol, right? These lipids are used as long-term source of energy, right? And they play a very important role in the composition of the plasma membrane. How about the proteins? They are going to play a structural role and a functional role. As functional role, we're going to have enzymes, we're going to have carriers, we're going to have hormones, we're going to have movement, right? Okay, so what are proteins typically used for? They are used as support, metabolic, transport, regulation, and motion. How about nucleic acid? Nucleic acids occur in 2 kinds, DNA and RNA. They're made up of building blocks called nucleic acids, and they are used to store the genetic information and also process that genetic information to produce proteins. What I did not say earlier, it's that the building blocks of proteins are called amino acids, and the building blocks of sugars are called glucose. Well, that's one kind, okay? You have many, you have galactose too, you have fructose, you have ribose, you have deoxyribose, to name only those. All right. So review the end of the chapters that we just covered. Okay. Scroll down to the end of the chapter, and you're going to see a review there. Please take the time to review the summary at the end of the chapter. Also, do some assessment questions. There are some assessment questions at the end of the chapter. There are also some assessment questions that you can design yourself, right? And finally, create handwritten concept map, just like before, right? Please do that. It's going to be helpful to yourself. Thank you for your attention.